Okay, let's start it with again, the most important session of the day, certainly the most exciting and more scientific. So that's, I don't know if you can turn on the session 2A, where we want to talk about regulatory science. And we want to have myself and two Davids, David Litvak and David Eberhard. Hi, David. So the problem with this session is too many Davids and no Goliath, but we will try to have some fun. So I'm Adrian Senderowicz. I'm the CMO of a company recently moved from Sanofi. And next slide, please. Oh, I have to move myself. Okay, sorry. So my disclaimer, so these are my views and that doesn't represent necessarily views of my company. So this is in general, regulatory science is a little bit dry. I'll try to make it as much fun as possible. But I think that there are some basic issues that I've been talking to different folks from academia and even industry that most folks do not understand, or they are not aware of this. And I think it will be useful. I want to talk more about the present and past of regulatory issues, and then David Litvak is going to talk about the future, hopefully, and beyond. And then David Eberhard is going to talk about some clinical utility. It's going to be fun. So my presentation is going to be divided in four aspects. I mean, who does what at the FDA? Many people say the FDA told me X. What does it mean, the FDA? It's like saying the government. Something happened in the government, and it's a very imprecise word. And I think that different folks in the FDA and even in different divisions think differently. So I think it's important to define that. What is an in vitro diagnostic, companion diagnostic, and we'll try to discuss and give you some examples. What is Codevelopment, and then a few examples that I think that people will understand better based on that. So who does what at the FDA? CDER, CBER versus CDRH. And so basically, the two centers that regulates drugs and biologics are CDER and CBER. In order for these are they regulate drugs and biologics, including gene therapy, vaccines, etc. And in order to be able to start a clinical trial in CDER or CBER, you need an IND, and when a drug or biologic is ready to be marketed, you submit the NDA or BLA, and I will go more in depth in the next slide. In contrast, CDRH, or Center for Devices and Radiological Health, that David belongs to that particular center, are responsible for approval of medical devices, and in order to start a clinical testing with a device, you need an IDE, and the approval process is for me, it's very complicated because I was in CDER in a sense, but there are two main ways of approving or marketing these devices: is the PMA and the 501(k). So as a simplification, again, you have CDER and CBER. When a sponsor or a company or even individual wants to talk to CDER and CBER, what you have is a pre-IND that you discuss the initial questions. For CDRH, it's called pre-submission. As I mentioned to you before, when you want to allow starting testing in humans for the CDER, it's called investigation new drug, and there are completely different regulations, and it's not that the name is different, it's completely different regulations. For the CDRH, it's called investigational device exemption, also known as IDE. And for the approval for marketing, as we mentioned, NDA for CDER, BLA for CBER, and pre-market approval for CDRH and 501(k). Today, I want to talk more about IDEs and pre-market approvals. So that's 101 regulatory, and let me discuss about the in vitro diagnostic, companion diagnostic. And an in vitro diagnostic product, this is the definition from the Code of Federal Regulations are reagents, instruments, and systems that are important for diagnosis disease in order to cure, mitigate, or treat or prevent disease, intended for use in the collection of specimens taken from the human body. So this is a very broad definition. And there is a specific in vitro diagnostic product that's called in vitro diagnostic companion diagnostic that is specific, and what it says is that this device is essential for a safe and effective use for a corresponding therapeutic product. So in order to use this therapeutic product, you need to use this particular in vitro diagnostic product. And because these interrelated, the labels are, and for the drugs and the in vitro diagnostic are referred to each other, and this is the guidance if you want to read more about this in vitro diagnostics. So I'll give you an example about co-development, that is the development of a pair between a drug product and a diagnostic device, between a drug and in vitro companion diagnostic. And if you see the parallel for a drug development, so you can see that in CDER or CDER, and this is the most known way or what most people know about the FDA. So a drug is being tested in the lab. At some point, the sponsor believes that can be tested in humans. You submit an IND. There is a clinical development that you have phase 1, 2, 3. Sometimes you can approve drugs in phase 1 or phase 2. And then you have an approval that is a NDA or BLA. However, as we mentioned for the last few hours, these days there is a significant interest in approving drugs based on in vitro diagnostics. And there is a parallel, I will say, pathway where you have an assay development. You want to understand what is the nature of the assay. You need to have a validation. And then you need to test the clinical validation. And the clinical validation happens in the same trial as the validation for the drug. And the whole idea is to have a contemporaneous or simultaneous approval. So the drug is approved because the in vitro diagnostic test is approved and vice versa. Yep. So again, these are the definitions for an IDE. It's a regulatory submission that permits the clinical investigation of a device. If you have an approved IDE, you can ship your device without complying with our requirements. So basically, in order to ship a device, you need to have a pre-market approval unless you have an IDE, and you can submit, you can send this interstate. And what is important is focus on risks. So the IVDs depends on the risk, and depend on the risk, you require an IDE or not. And even though an IVD may not be high risk at the beginning of the testing, depending on the clinical situation, an IVD may become a high risk IDE. And there are some delegated responsibilities, particularly for IRBs, to enforce this particular issue. So these are what are the main features for the labeling of the IVDCD. The one important, very important issue is the analytical validity. What are we measuring? Are we measuring this correctly? What is the reproducibility, the sensitivity, the specificity? And something that is crucial, and I think that Bruce mentioned in his lecture, is the cutoff. Because not only it has to be positive in some occasions, but has to be positive in an X number of cells or percentage of cells. So, for example, for the ALK testing, you need to have a positive means more than 15% of positive cells. If it's 14, or if it's 13 or 10, the sponsor defined it that was negative, and those patients were not included in the trial, so you don't know what kind of data you will have for patients who are negative based on the cutoff. I mentioned about the risk and the context. Are you going to use this IVD in the clinical laboratory? Are you going to use it as in the outpatient setting? And what is the clinical purpose and the target population? Is it going to be used for diagnosis, for prognosis, for monitoring, or for stratification factor? And what is the target? Is it going to be in colorectal cancer? Is it going to be in KRAS mutant colorectal patients, metastatic, adjuvant, etc.? So this is important features in the IVDCD labeling. And this is an important issue when we say about risks. And the risks are related to the basically risks and benefits of having these tests in patients. And this is associated with the false positive and false negatives for this test. So as you can imagine, a false positive test, a patient will receive a therapy. And as you know, drugs, oncological drugs have significant toxicities. We are dosing at the very high doses. And some patients may receive a drug that unnecessarily that may be causing toxicities without any benefit. On the contrast, there is the risk of false negatives. That means the patient would need to receive that drug, but because it's a false negative, the patient will not receive. And those are risks that the CDRH considers when they define the risk for this device. And the policy, so they divide, and this is a simplified way of saying, you have devices that have significant risks, and devices that have significant risks need an IDE submitted before testing these IVDs, yes. And there are others that are considered non-significant risk. So an IDE is not required. However, there are some minimal requirements that need to be submitted, and so it's called abbreviated requirements. And what people recommend, the pre-sub, so initial interaction with the CDRH is recommended. So a long definition and the CFR, and I apologize, but to make a long story short, how do you define what is significant risks? Is when this IVD is important for patient care. So if you're going to select a patient, so if you define this patient have these characteristics that will lead to the treatment with Y, and that in general is a high risk that requires an ID. So another issue that several folks brought to have some interesting discussions, and again, I'm not here to side with one side or the other, but I think it's important to state that there are issues regarding laboratory diagnostic tests. It's a very important, it's very important for research. Even most of the in vitro diagnostic tests, companion diagnostics, started as a laboratory diagnostic test. As basically Bruce mentioned, the EGFR and the ALK fish started as an LDT, and eventually, and as I will share in the next few slides, evolved as a companion diagnostic and as a commercial test. And the definition is a type of IVD that is designed, manufactured, and used within a single laboratory. And that's the way that the FDA defines. And LDTs measure a wide variety of analytes. For example, an LDT will measure glucose, cholesterol, DNA in a sample. And they obviously, with science, have evolved, so LDTs have also evolved. And these days, some of the IVDs have similar risk as the non-IVDs. And the other important issue that the FDA does not consider a diagnostic device as an LDT if it was designed or manufactured completely or partially outside the laboratory that offers us uses them. So this is important because it will have some consequences in the near future. And as it was mentioned by one of the speakers, as of today, I would say, FDA has not enforced pre-market review of these LDTs? Because this regulation started in 1976, and most of these LDTs were simple and were low risk or non-significant risk. But things evolve, are becoming more complex, and present higher risks. And in July, the FDA submitted a letter to Congress with some proposals to regulate some of these LDTs. And these are the reasons why they believe it's important. And I'm not defending again the FDA, I'm bringing the facts. And now I think that there is in the 120-day process where sponsors and people are submitting comments to this potential legislation. The FDA found faulty LDTs that led in patients with heart disease under and over treatment, in patients with cancer the same, and even found patients with incorrect diagnosis of autism based on LDT. So it's a serious issue, and there are some proposals from the agency, and I would, and probably David is gonna discuss more in detail, but I foresee that it's gonna be significant changes in the legislation for LDTs. So the examples, so I think that the godfather for the companion diagnostics was in 1998, Herceptin, or also known as trastuzumab, and the initial test was DACO to try to measure HER2/neu. And as some of, I don't remember who presented some data, I think I remember in the table discussion, the HER2 is not as simple as we thought, and there are multiple different tests approved by the agency in order to determine whether the patient has HER2 positive for breast cancer. So certainly at the beginning, the data, so basically the science knew as much as was available, and despite that, that test predicted which patients will respond or not. However, there was some refinement over the last few years. And let me, in order to finalize my presentation, I'll give a presentation for a drug called Crizotinib. This, in part, was presented a couple of years ago at the Institute of Medicine. I will give my own views about it. And I think it's a good example to understand the companion diagnostic issue, and also that this could be done even for a big pharmaceutical company as Pfizer, that they did it in record time. So anybody can do it, I think. So this is a small molecule tyrosine kinase inhibitor that inhibits several tyrosine kinases, but more potently the c-Met and the L-tyrosine kinase. In 2006, the phase 1 trial started. At the recommended phase 2 dose, there was a significant enrichment for c-Met patients, and with a variety of LDTs. Unfortunately, there was not much activity in that initial enrichment. However, there was a discovery from a Japanese group that there was clear evidence of a translocation between EML4 and ALK gene. This translocation is an oncogene driver. So people in Mass General put the first patient based on an LDT, a fish LDT, that and demonstrate significant response, and basically based on this clinical data that trumps anything that people think from the laboratory, they changed the clinical development plan, and they started enriching based on ALK patients, ALK positive based on this fish. And in 2011, the FDA approved this agent based on Subpart H, also known as accelerated approval or conditional approval. And in 2013 was converted to full approval. So let me give you the timelines from the drug point of view, and then in the next slide I will give you the timelines from the companion diagnostic point of view. So as I mentioned, 2006, the phase 1 started. 2007, that particular patient with a translocation have a significant response, significant change in the development of this agent, multiple publications, and then in phase 2 and two phase 2s, single arm studies, and one phase 3 randomized trial was started And in 2011, the drug was approved by accelerated approval, and in 2013 was approved for full approval based on increase in PFS over chemotherapy. From the CDX development, the same timeline, as I mentioned in 2007, based on this publication, there was a FISH LDT created in Mass General that in collaboration with Abbott and Pfizer, they basically have a new FISH assay that's actually very similar to the Mass General one. There was an IDE before the pivotal studies. It was locked. So this is very important that when you're going to start a registration or pivotal study, to lock your assay, because you cannot change in the middle of the pivotal study your test. And basically have an approval of Crizotinib and the Vice's Albrecht part test at the same time. It was a simultaneous approval. So after this brief introduction to a little dry, I would say, regulatory presentation, I would say that when you have a drug, when you're working with a drug that requires an IVD, you need to start early thinking about the companion diagnostic. You need to talk to the CDRH, not only to CDER, to CDRH, to try to submit the IDE as soon as possible. The IDE depends on the risks, and certainly when you start selecting patients based on in companion diagnostic, you need to have an IDE in place. As I mentioned to you, if you're planning to do a registration trial, you need to have a lock IVD in place. As I mentioned to you, the LDT regulatory landscape is evolving, and it will be evolving, I assume, in the next few years. And I also mentioned that safety and efficacy of the new drug and the clinical utility of the new diagnostic happens in the same trial. And the goal of co-development is a simultaneous approval of the IDE and the drug. So these are some of the recommended reading, and thank you. So now I want to ask David to present his presentation. Get Nancy and